Good afternoon. Thank you for joining us for Research 411, a talk show focused on sharing best practices and trending research topics delivered by professional staff and prominent UT Dallas researchers from various disciplines. I am Tiffany Willoughby, Assistant Director in the Office of Research and Innovation. I will serve as a session moderator for today's discussion. During today's talk show, Dr. Shashank Sirsi will discuss his research on the treatment of neuroblastoma cancer. Dr. Sirsi grew up in California and attended the University of California, San Diego, where he majored in biomedical engineering. As an undergraduate, Dr. Sirsi performed research in a physiology lab in the Veterans Hospital, where he studied the biomechanics of single muscle fibers. Dr. Sirsi graduated with a Bachelor's of Science in 2002 and continued his research at Drexel University, where he designed novel polymer vectors for nucleic acid delivery to dystrophic muscle tissue. He received his Ph.D. in Biomedical Engineering in 2007 and gravitated towards cancer research during his first postdoctoral position at Columbia University in New York. As a postdoctoral researcher, Dr. Sirsi designed and tested novel ultrasound contrast agents for in vivo imaging and targeted drug delivery to tumors using pediatric neuroblastoma models. In 2010, Dr. Sirsi moved to the University of Colorado at Boulder and continued his cancer work as a research associate. He was awarded an NIH R21 grant in 2012 to support his research on image-guided drug delivery to tumor tissue. He subsequently worked as a senior research scientist at MIT and the ETH University in Switzerland, using ultrasound contrast agents for MRI-guided focused ultrasound drug delivery to the brain. During his academic tenure, Dr. Sirsi has published over 50 research articles, 2 book chapters, and has been awarded multiple patents. Currently, Dr. Sirsi is an assistant professor in bioengineering at UT Dallas. Thanks for joining us today, Dr. Sirsi.
Okay, thank you for the introduction, Tiffany.
You are most welcome. Can we start off today by having you tell us a little bit about your research?
Sure. So here at UT Dallas, I run the image-guided drug delivery lab, or more recently termed it the Acoustic Neuroblastoma Therapy Lab. Our research is predominantly focused on a pediatric form of cancer called neuroblastoma, and we are trying to use sound waves to sensitize these tumors to different forms of therapy. Primarily, we're interested in delivering nanoparticles, nanoparticle drugs to tumor tissue. Using these sound waves, we engineer sound-sensitive particles, which are essentially small gas bubbles. These bubbles will vibrate in an ultrasound field. So with judicious application of the ultrasound energy, so the sound waves can travel through the body to these particles which are circulating in the blood, and you can trigger release of drugs that are encapsulated in the bubble, or you can use the mechanical oscillation, the vibration, to permeate tumor tissue and get drugs to extravasate from circulation more easily into the solid tumors. So we are developing novel strategies on all different aspects of this research, from the imaging side to detecting the bubbles and using the imaging or perfusion of the tumors to predict drug uptake in tumors, to developing particles that are can carry a lot of drug and a lot of chemotherapeutic or other therapeutic agents and get them into the tumor tissue.
Thank you very much. Can we maybe take a step back and identify more specifically what is neuroblastoma? And not only that, what are the benefits of the new therapies that you are developing in relationship to the current therapies that are currently being utilized?
Got it. Yeah. So neuroblastoma is a aggressive, the form of neuroblastoma that we study is an aggressive pediatric cancer. It's the most common extracranial solid cancer that's found in children. So all childhood cancers are rare, but this is one of the more frequent of the rare cancers. Neuroblastoma, it's often confused as a brain tumor. It's not a brain tumor. It's actually an abdominal tumor. It forms in the typically the adrenal gland in the kidney. It's derived from neural crest tissue, but it presents in the abdomen, from there it will grow and spread. We are so neuroblastoma itself is about affects about 1 in 8,000 children that are born. It's a very rare form of cancer, but it is a very aggressive form. It is very deadly. Treatment of neuroblastoma typically requires lots of different therapies: systemic therapies, chemotherapies, targeted radiotherapy. And one of the major problems, especially with neuroblastoma, is the therapy itself. Yeah, you can imagine these children are these are infants that are, they're typically diagnosed around 1.5 to 1 year old. They are growing. All of their tissue is developing, and at this time they have to receive these kind of toxic treatments that are designed to kill cancerous tissue. You can imagine that this is going to have some effect on the healthy tissue as well. And this is why our lab is particularly focused on pediatric cancers, because we want to try to eliminate some of these toxic kinds of therapies and replace them with more rational targeted therapies, especially using sound waves, which are non-ionizing forms of energy compared to radiotherapy. And we want to try to achieve better results with less off-target effects.
That makes sense. And I know you highlighted initially a little bit about how the sound waves work. Can you kind of break it down for our audience in a step-by-step scenario so that it can paint a picture in their heads of what actually takes place with your therapy?
Sure. So essentially what we design in our lab are sound sensitive particles, or gas bubbles. We take a hydrophobic gas and we encapsulate it with a biocompatible material, lipids, polymers, or proteins. Typically we use lipid encapsulated bubbles. So these bubbles, because of the compressibility of the gas core, they're able to respond to sound waves. So sound waves are essentially oscillating pressure waves. So just simple Boyle's law: when the pressure outside of the bubble drops, the bubble expands. When the pressure outside the bubble increases, the bubble shrinks. So you get this oscillation or mechanical vibration in response to a high frequency, about 1 to 10 megahertz sound wave. So this bubble is essentially growing and shrinking 1,000,000 times per second when it's in a sound wave, 1 million to 10 million times per second, depending on the frequency of sound that you're applying. So this is extremely rapid. You can't see this happening. Well, not with a conventional camera. There are very, very high speed cameras which you can use to actually see this happening. But this mechanical vibration, it creates a lot of force. And the goal of our lab is to, how do we best utilize that force of this vibration to promote a therapy, promote a release of a encapsulated drug, for example, or use that force to permeabilize, to open up spaces within the vasculature of the tissue. So the drugs used to treat these cancers, they are typically introduced into the bloodstream. So if you introduce the bubbles with the therapy in the bloodstream, they'll circulate throughout the body. And if you apply the ultrasound to the tumorous regions or to the tumor itself, you can try to permeabilize the blood vessels. And when you make the blood vessels more permeable, it allows the drug to come out more easily and treat the cancer tissue where it needs to go.
Okay, so does this in fact take place concurrently then with chemotherapy, or
Sorry, go ahead.
That's okay.
Yeah, currently the major focus of our lab is to systemically deliver the bubbles with a special form of chemotherapy called liposomal doxorubicin. So liposomal doxorubicin, it's sold commercially. Doxorubicin is a very small molecule. It's probably the most common chemotherapeutic used in cancer. It is a poison. It intercalates with DNA. It prevents cell division, transcription, translation. So that's how it it functions. It's a very small molecule, so it actually goes everywhere in the body when you inject it into the bloodstream. This is why patients undergoing chemotherapy will typically also lose hair, fingernails. You're stopping all growing tissue in the body. So this we haven't developed this technology. The nanoparticle, the it's called liposomal doxorubicin. They've taken this small molecule drug, they've put it into the core of a small 200 nanometer particle. And this particle will circulate in the blood for a very long time and prevent the drug from getting into areas where it can cause damage. So specifically the heart. Doxorubicin causes a lot of cardiotoxicity in patients. It's dose limited in that sense. We're trying to take this small particle that circulates very well in the bloodstream, and we're trying to get it to preferentially extravasate or accumulate in tumor tissue. So by encapsulation of this drug and then release of this drug into the tumor, you're preventing that poison from getting to other areas of the body.
And then, therefore, reducing any side effects of the treatment.
That is the hope, yes. More of the drug to the tumor, less of it to the heart and to other areas where it's toxic.
Perfect. Now, what led you, or what is your drive behind conducting this type of research?
So I am an engineer, so I've been very much on the materials side of things ever since I was in graduate school. So I focus on developing polymers, more on the physics of how this gas particle works. So creating these gas particles, engineering these gas particles. Really, these are kind of agnostic tools. It doesn't care. The particle doesn't care what tissue it's in or where the drug is going. That's really up to us as researchers to find a particular disease model which we want to treat. I really got into neuroblastoma research, neuroblastoma and other pediatric forms of cancer, but primarily neuroblastoma, because I was working at Columbia University. I was a postdoctoral researcher in a lab that was developing novel lipid bubbles. And these researchers, they didn't actually want to use these bubbles for drug delivery. They wanted to use it to image. So these bubbles actually are traditionally used for imaging. The vibration of the particle generates a sound wave. You can see it on an ultrasound scanner. Traditionally, what these particles are used for are visualizing the vasculature of tissue or tumors. So I worked with a pediatric oncologist at Columbia University, pediatric surgeon, pediatric oncologist that I still work with today. And they were very interested in mapping the vasculature of these tumors to see if the tumors were responding to different types of drugs, so specifically anti-VEGF or VEGF blockade therapy. The idea is that if you map the vasculature of the tumor and you watch how the blood vessels are changing in a tumor, that happens more quickly than the actual change in size of the tumor. So you can, if the drug is not working, you can find out very quickly using ultrasound imaging and move on to a different drug, getting rapid feedback in order to assess whether a therapy is working or not. So that's what this group was very interested in. Now I kind of merged that idea, mapping the vasculature of tumors, with drug delivery. So we're imaging these tumors. Why not at the same time deliver a drug? And that was what my first grant was kind of based off. But really, my involvement with neuroblastoma was thanks to these other physicians, oncologists, and vascular biologists that have been working in the neuroblastoma field for their entire lives.
It sounds very interesting and inspirational, and it's good to hear that you still work with them as well on such a good cause and excellent research. At this stage, do you feel like the drug delivery to the tumors is precise? And what types of outcomes have you seen with this to date?
So again, my research is in uh, preclinical research. So we are testing. We're not testing this in humans in our lab. We do have orthotopic xenograft tumor models, which we do test this on, and the technology itself is very effective. We can show we've published this a year ago. We have just a recent publication that we just submitted to Theranostics, which is a very high impact drug delivery journal, showing that we can get liposomal doxorubicin to neuroblastoma much more effectively using this sound sensitive approach. Um, uh, the the issues kind of come in with, okay, how do we translate that to human testing? So we know the strategy can work in a mouse model. Now we're thinking about what are the strategies now. How do we get this to work in a human? That's kind of like the next big leap that we're kind of focusing on right now. So really, this I think that was a long-winded answer to your question. This is not something that's in clinical trials for neuroblastoma. There are other cancers, glioblastomas, for example, pancreatic cancers. So this where other research groups have developed this technology to the point where it is in limited human trial testing, or probably more advanced testing at this point. But in terms of pediatric cancers in neuroblastoma patients, this is not a technology that's in clinical practice yet.
Thank you, Dr. Sirsi. Now, in your interview with the Dallas Morning News, and we'll drop that article in the chat for the audience members to view, you mentioned that since your son has been diagnosed, you've been more clinically focused on your research. What about the clinical aspects of your decade-long study into neuroblastomas really surprised you the most?
Yeah, so that's a great question. My son, he is a little over 2 years old right now. He was diagnosed with neuroblastoma in 2020. So I've been working in neuroblastoma for over 10 years, and like I mentioned to you earlier, that my focus had always been much more on the engineering, on the material side of things. How do we generate these particles, test them in a laboratory, in solution, and then it always had been less focused on how do you apply this in a clinical setting. So I've been working with neuroblastoma for more than 10 years, but my son was the first patient that I had met with neuroblastoma. And that was the first time I really understood what it was like for neuroblastoma patients to, or parents of neuroblastoma patients. Remember, these are children, 1, 2 years old. They're not going to remember a lot of it, but the parents, what they have to go through, how a chemotherapy regimen actually works in a clinical setting. So these are things that I just honestly had not thought much about. Everything was more abstract to me. Everything was more theoretical, mathematical. That now, I have a much more broader, deeper understanding of what a treatment looks like, what a patient actually goes through, and that has been something that has been um, I would say infiltrating into our future research focus in our lab.
Absolutely. How has, in fact, being your son's parents affected you and your wife's view of children suffering from this disease? And how then do you communicate it to others, whether it be other, you know, parents of families that you know who have children with a neuroblastoma, or just others in the community?
So this is something that I have, my wife and I have discussed this back and forth. How much of this is really our son's story, right? He, yeah, as much as it is our story. How much of his medical issues do we disclose? We really kind of came to the conclusion that, you know, we want to promote awareness of neuroblastoma, and you know, and we didn't honestly, when he was going through chemotherapy, he his prognosis is excellent, by the way. I don't want to try to mislead anyone right now. He's doing very well, but he did have to go through chemotherapy, and there was, you know, quite a bit of time we were unsure of what was happening, and during that time we didn't really want to talk about it that much. It just really, you know, when we, you know, found out his prognosis was going to be excellent, or as good as it can be for having cancer, decided to be a bit more open about it, and really, I've shared his story, our story, with people in my class. So I teach an image-guided drug delivery class. Usually in talks now, I insert my son into the talks to you know, talk about the, you know, and I discuss openly that, you know, this is you know, something very personal to me now. It didn't feel, I was always interested in this before, but it didn't feel personal before. Now it's much more personal.
Absolutely. And I can imagine so. Thank you for all that you do for that community, your son and everyone else who is touched by this cancer. So we appreciate you and everything that you do. We do have a few questions from the audience, and they are related to microbubbles. Where do they come from? How do you test them? How are the microbubbles able to release on command, so to speak? What are they made of? And again, if you could reiterate exactly how they work.
Sure. So I'll go over the microbubbles are small gas bubbles. They're predominantly a gas core, usually a hydrophobic gas, perfluorobutanes, what we use in our lab. In order to make them, we emulsify the hydrophobic gas with a surfactant in water. So the surfactant is either a phospholipid, a protein, or a polymer. So predominantly, we use phospholipids. These are the same molecules that comprise the cells that comprise the outer layer of cells, the cell membrane. So they're highly biocompatible, and the gas itself can actually be exchanged out in the lung. So these are completely biocompatible molecules. The nature of the surfactant is that it's amphiphilic. It'll have a hydrophobic and a hydrophilic portion. So the hydrophobic tail of the phospholipid will orient towards the gas. The hydrophilic head will orient towards the water, and it forms a monolayer, a very thin, couple nanometer thick, flexible shell. So you can imagine this is like, you know, the outer, you know, the same material that comprises a cell, but instead of all the stuff in the cell, you just have a gas. So we have equipment in our lab that we can use. We have to use a very high power emulsification technique, a probe tip sonicator, that will mix the gas and the water and the surfactant all together, and then we form stable bubbles. So the purpose of that shell material is to stabilize the gas bubbles. So gas bubbles, free gas bubbles, will tend to coalesce, ripen, and grow uncontrollably in close proximity. So the shell stabilizes them and prevents that from happening. So you have these stable bubbles, but still the shell is very flexible. So the gas can expand and the gas can shrink during an oscillating pressure wave, which is a sound wave. So the bubbles will grow and shrink when you apply ultrasound to them, and it's that mechanical agitation, which is what we're using to release an encapsulated drug or mechanically alter the vasculature of a tumor. The way that we do drug release, now there's a few different ways that we do this, but one of the ways is that we can take an encapsulated drug and chemically bind it to the surface of the bubble. So then when the bubble is vibrating, oscillating, the mechanical forces are translated to the the surface bound or tethered particle. And the idea is to use those forces, and we have a few different techniques that we're doing this, different ways of conjugating the bubbles and the drug carriers together. The idea is to rip apart the drug carrier and release the drug so that it can freely diffuse into the tumor from the blood. So you apply the ultrasound only where you want these particles to release. And the idea is that everywhere else, they'll stay encapsulated and they'll be less toxic.
Dr. Sirsi, that makes perfect sense. And I can visualize exactly what you just said. And also with the previous knowledge related to what I'm going to call little people with shields on already circulating through the body to protect the rest of the cells and the organs. So thank you very much for painting such a clear picture. I just want to remind the audience that we are open to additional questions. If you have them for Dr. Sirsi, now is your chance to ask them. And it does look like we have one, Dr. Sirsi. How, let's see here, US and microbubbles help increase permeability around the tumor in the vasculature. So I think that they're asking, how do
Ultrasound?
Yes, ultrasound
Okay.
and microbubbles help increase permeability around the tumor in the vasculature.
So really, it is the vibration of this particle. So this is traditionally how these bubbles operate, and they're used in drug delivery applications. The idea is that the bubbles will grow and they'll shrink. When they grow, they can stretch the vascular endothelium, but it's really when they shrink, the vascular endothelium is able to accommodate high pressures. It's used to accommodating blood pressure, for example. It's not used to that negative pressure. So water is incompressible. So when the bubble shrinks, it pulls in its surrounding. And that's when it causes vascular endothelial cells to delaminate. It basically pulls the endothelial cells apart using by forcing it in a way that it's not accustomed to. So this is not something that our lab developed. This is something that's, this technique has been studied by a lot of other research groups over the last 20 years. What we focus on is the design of the particle itself. How do we best put a drug on or change the size of a particle? How do we manipulate the physical features of the bubble specifically for these drug delivery applications?
That makes sense. And when you say delaminate, I visualize a layer of a cell kind of breaking down and breaking apart. Does that make
Yeah,
sense? Is that what you're
exactly.
meaning?
Yeah,
Okay.
so the vascular endothelial cells will become delaminated, but they'll detach from the extracellular matrix. They'll become disjointed. The gaps in between them will increase, and there are other mechanisms by which the bubbles can promote drug uptake. This is one of the more commonly thought mechanisms to occur. And this is temporary, so that they'll fix themselves over time. So really, you're opening up the blood vessels for a transient amount of time, and then you let it rebuild after the therapy has been administered.
Very good. Another question from the audience in the queue. Is it important to have homogeneous bubbles in case of their size? Thank you.
Yes, I think someone might be already familiar with some of the research. I believe, yes, that is this is a very technical aspect of the field that not a lot of people pay attention to, but the response of a bubble is very dependent on the size. So a bubble of 5 microns and a bubble of 1 micron they're going to respond to sound waves differently. And each of these bubbles have what are called a resonant frequency. Honestly, the best way to utilize these bubbles is to activate them at their resonant frequency. And if they're all the same size, you can use one frequency of sound to activate all of the particles. Otherwise, you have different size bubbles. Their overall uh, fundamental reaction is harder to predict. So one of the actually some of the sponsored research that we're working on right now with a colleague of mine, he's developed a method of generating microbubbles of a very specific size. He's kind of optimized that size for drug delivery application, and we're testing that in our lab right now using neuroblastoma tumor model. So that's an excellent question.
All right, Dr. Sirsi, and I don't have any additional questions in the queue. I want to thank you again so much for your time today. We appreciate you and everything that you do for our community. So thank you again for joining us today, Dr. Sirsi.
Okay, not a problem. Thank you for inviting me and hosting me.
Absolutely. And audience, thank you for attending our program today. If you enjoyed today's talk show, please suggest it to a friend. To stay abreast of Office of Research and Innovation information and programming, please follow us on Instagram and Twitter and subscribe to our newsletter. Feel free to use your phone to scan the QR code on the slide or follow the links that are posted in the chat. And finally, if you have a moment, please take time to complete our research event survey. We look forward to having you join us for an upcoming Office of Research and Innovation event. Thank you and have a nice afternoon.